Hello everyone. This video provides a comprehensive understanding about glucose, its homeostasis, clinical significance, and the analytical methods used to measure it. Glucose is the most important carbohydrate which is used as the primary source of energy in the body. Although a small amount of glucose is found in the diet in its monosaccharide form, the majority is found in the complex carbohydrates in the form of polysaccharides and disaccharides. Glucose homeostasis is maintained by various hormones, primarily through the actions of insulin and glucagon, which are produced by the pancreatic beta cells and alpha cells, respectively. These endocrine hormones are secreted in response to external signals such as nutrient intake, stress, via humoral, neural, or hormonal signaling pathways. Upon consumption of food, the circulating glucose is taken up by the facilitative glucose transporter 2 or GLUT2 located on the surface of the beta cells. Once inside the cells, glucose undergoes glycolysis, which leads to the production of adenosine triphosphate or ATP. This results in the closure of ATP-sensitive potassium channels, which are open under non-stimulated conditions to maintain the resting membrane potential. However, upon closure of these channels, the decreased outwardly potassium current and the opening of voltage-dependent calcium channels elicit depolarization of the cell membrane. The subsequent increased intracellular calcium triggers the fusion of insulin-containing granules with the cell membrane and thus release of insulin into the bloodstream. As can be seen in the upper part of this diagram, insulin then interacts with its receptors on muscle and adipose tissues that results in the uptake of glucose by the insulin-dependent cells, thereby decreasing the exogenous glucose concentration from the circulation. Furthermore, insulin promotes glycogenesis, which is the process of storing glucose in the form of glycogen, and lipogenesis, which is the process of synthesizing fatty acids. In contrast, glucagon is secreted by the pancreatic alpha cells in response to hypoglycemia, prolonged fasting, exercise, or protein-rich meals. The mechanism involves uptake of glucose via glucose transporter 1 or GLUT1 in the pancreatic alpha cells, leading to glucose glycolysis and subsequent generation of ATP in the mitochondria of the alpha cells. Via this mechanism, ATP generation in the mitochondria of alpha cells reflects the circulating glucose concentration in the blood. Hypoglycemia results in decreased ATP production, which leads to the closure of ATP-sensitive potassium channels, opening of voltage-dependent calcium channels, and depolarization of the cell membrane. As a result, the increased intracellular calcium triggers exocytosis of glucagon-containing granules and release of glucagon in the circulation. As can be seen in the bottom half of the diagram, the circulating glucagon interacts with its receptors located in various tissues, but primarily in the liver. It promotes gluconeogenesis, which is the breakdown of glycogen and release of stored glucose into the circulation, as well as inhibits glycogenesis to reduce glucose storage. Glucagon also stimulates the breakdown of fatty acids and amino acids to increase the production of non-carbohydrate energy sources. In summary, the opposing actions of insulin and glucagon maintain glucose homeostasis. Glucose test is requested primarily for the diagnosis of diabetes, but also in other conditions such as insulin resistance, impaired beta cell function, reactive hypoglycemia, acromegaly, and other disorders of carbohydrate metabolism. Initially, glucose was tested in urine samples, but due to its inaccuracy as urine glucose does not reflect glucose concentration in the blood, it's now commonly tested using blood samples. Although glucose concentration is highest in arterial circulation, laboratory measurements of glucose is performed on all types of samples, including capillary, whole blood, plasma, serum, urine, pleural fluid, acidic fluid, and cerebrospinal fluid. Whilst fasting glucose concentration is similar between venous and capillary sample, postprandial and glucose measured as part of oral glucose tolerance testing differ significantly between these two sample types. As a result, it's recommended that capillary samples should not be used to diagnose diabetes in non-pregnant adults. Glucose can be measured on a random or fasting blood sample as part of tolerance tests to assess insulin resistance or screen for gestational diabetes, and by measuring the level of glycated hemoglobin A1C to monitor glycemic control. Whilst there is almost no challenge in the analytical aspect of glucose testing, as laboratory methods have been refined to have very low analytical variability, there are many factors that can affect the glucose concentration in the sample in the pre-analytical phase. These factors include sample collection techniques, sample type, transport conditions, delayed sample processing, and absence or presence of additives that inhibit in vitro glycolysis. The most important pre-analytical aspect to minimize artifactual glucose result is to prevent in vitro glycolysis caused by the glycolytic enzymes present in leukocytes and erythrocytes. Enzymes involved in the preparatory phase of glycolysis include hexokinase, phosphohexose isomerase, phosphofructokinase 1, aldolase, triose phosphate isomerase, and in the payoff phase, enzymes such as glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate mutase, enolase, and pyruvate kinase are involved. There are two ways to prevent or reduce in vitro glycolysis. Number 1. Placing the sample tube on ice immediately after collection and separating plasma from the cells using centrifugation. Number 2. Use of additives to inhibit the activity of the glycolytic enzymes. The first method is technically not practical, especially in distant phlebotomy sites. Therefore, the alternative method, which is the use of additives such as iodoacetate, D-mannose, sodium fluoride, and citrate buffer in the collection tube, is now commonly used. Iodoacetate inactivates glyceraldehyde 3-phosphate dehydrogenase. It inhibits glycolysis in the payoff phase, which means that glucose will still be consumed in the preparatory phase. D-mannose is a competitive hexokinase inhibitor. However, the inhibitory activity does not last very long. It also interferes with glucose oxidase and hexokinase analytical methods. Sodium fluoride inhibits enolase by forming a fluorophosphate ion binding to magnesium, which is a cofactor for enolase. However, this does not stop glycolysis in the first 2 hours. The studies have shown that use of buffer such as citrate to lower the blood pH is quite effective at inhibiting glycolysis. Acidification of blood to pH 5.3 and 5.9 immediately inhibits hexokinase and phosphofructokinase enzymes, thereby instantly stopping glycolysis. Things to consider for this is dilution factor and the glucose results, because cutoffs are based on sodium fluoride sample types. There are 4 different analytical approaches to determine glucose concentration in the laboratory. These approaches include reducing methods, aromatic amine condensation methods, enzymatic methods, and electrochemical methods. There are multiple variations for each method. The most common analytical methods are based on using enzymes such as hexokinase, glucose oxidase, and glucose dehydrogenase. The hexokinase method is based on a coupled enzyme that uses hexokinase and glucose 6-phosphate dehydrogenase. It involves phosphorylation of glucose to glucose 6-phosphate by ATP in the presence of hexokinase and magnesium, which is then oxidized to 6-phosphogluconate by glucose 6-phosphate dehydrogenase in the presence of NADP+ or NAD+. This reaction produces reduced NADPH or NADH, which can be measured by absorbance spectrophotometry. The absorbance is directly proportional to the amount of glucose present in the sample. The glucose oxidase method involves oxidation of glucose by glucose oxidase in the presence of water and oxygen to gluconic acid and hydrogen peroxide. The hydrogen peroxide formed oxidatively coupled with a chromogenic oxygen receptor dye results in the formation of a colored compound that is measured calorimetrically. The color intensity is directly proportional to the concentration of glucose in the specimen. Glucose dehydrogenase method involves the oxidation of glucose by glucose dehydrogenase in the presence of a cofactor such as NAD+ to gluconolactone and NADH. The NADH produced can be measured amperometrically or calorimetrically. That's it for this video. Thank you for watching. If you've gained any value from this video, consider subscribing to the channel, give a thumbs up, comment for constructive discussion, and share the video with people who may benefit from the content. Thank you again.